Hi, I'm Chris,
And I'm Terry.
and we're back for the second lecture in the Nengo Summer School series. And what we're going to talk about today is really getting your hands-on with Nengo. So the last lecture, we sort of showed you some of the things people have done in the past, great big models people built, robotics, neuromorphics, and all kinds of stuff. And today we're going to dive in to give you a little bit more background about how we use the neural engineering framework in Nengo and basically give you tutorials so you can follow through and on your own computer, hopefully build up a couple of examples to really get a feel for how Nengo works. So as I just mentioned, the purpose of the lecture today really is to get your hands dirty, as it were, using Nengo. But I think it's helpful to start with a little bit of theoretical background and how people have approached modeling the mind or the brain in general. So historically, there have been three central views. The first, called good old-fashioned AI a lot of the time, or symbolicism, is really the idea that the mind is a computer, hence the picture there. And the main techniques used here are essentially language-like representations, if-then kinds of statements, and trying to describe high-level cognition often using those sorts of techniques. Production systems in particular, where you have something called a production, which is essentially an if-then rule. If it's a cat, then it's fuzzy. You can use those sorts of rules based on the input. So if you see a cat, then you might infer that it's going to be fuzzy. And you can actually do some very interesting and sophisticated sort of mind modeling using these sorts of techniques. Traditional methods along these lines are ACT-R and SOAR. These are sort of modeling approaches which embrace this technique, and they actually combine others nowadays with that. But really the core of those methods is to try to understand how we can explain human planning, human reasoning in a way which is sort of understandable linguistically using those kinds of representations. Now, that was a very successful way of approaching mind modeling to start. Basically in the 1950s and 60s and 70s, most AI and models of the mind were really taking this kind of symbolic view. But in the 70s, there sort of became a contender, a second contender, which is a connectionist view, or as we now know them, neural networks. This sort of burst on the scene and began to argue that symbolicist approaches, while they were good for certain kinds of tasks, like maybe planning your courses that you needed to take for the semester, or explaining some linguistic features or logical reasoning, which of course people undertake, it didn't really address a lot of other kinds of behaviors people exhibited and other animals, things like being able to perceive animals in noisy visual images, for instance. So one of the standard examples is chicken sexing. So when you see a little chick, right, they tend to all look the same, the little yellow fuzzy things. And experts who actually come to know chicks really well can look at the chick and determine whether it's a male or female with really high probability, but they can't state for you exactly what features they're using to do that kind of classification. So this sort of pattern matching, which is difficult to put into words or symbols, is something that connectionism really excelled at. It also introduced the notion of learning as being pretty fundamental to how the models were constructed. And this entire approach, also called PDP, or parallel distributed processing, with people like Jeff Hinton and Jay McClelland and David Rumelhart and Paul Smolensky and Terry Sinofsky, all of these names, which are now sort of like leading lights in the field, embraced this kind of approach and thought that it really had something interesting to offer in contrast to the standard symbolicist view. And so people often think nowadays that these are really the main two perspectives that people have taken on building minds, and that's largely because there are proponents of both of these approaches still, and there's arguments that go back to the 50s being rehashed nowadays between people doing deep learning and people looking more at sort of linguistic kind of behaviors. But there's another perspective that showed up in the 1990s called dynamicism. And I think it has something really interesting to add to the conversation, although it's often overlooked. And dynamicism really wanted to argue strongly that both symbolicism and connectionism are missing something fundamental about mental systems. And that is that these systems are embedded in a physical environment, and that in virtue of being embedded in a physical environment, they also are essentially temporal. So you can't really sort of extract time varyingness out of minds and still have a mind. Now, both symbolicism and connectionism have ways of dealing with time. So for instance, in the symbolic view, people often thought that, when a production is used, it takes about 50 milliseconds for the mind to sort of use that production. In connectionism, when you sort of recurrently connect neural networks back onto themselves, you sort of implicitly introduce a notion of time where you have to sort of update the network, and then at the next time step, you can propagate its own activity back to itself and update it again. And you have some kind of time step, but that doesn't really map to real time. So like, does one of those time steps take 50 milliseconds or 1 millisecond or 1,000 milliseconds? It's sort of, there's no standard. Dynamicists, in contrast, said, when you model a system like the mind, you should be modeling things in real time. So you always need to talk about time. It's not something that is sort of secondary. And really, time is a major determiner of whether a biological system is successful. So if I am walking through the forest and I see a tiger, and it takes me 100 milliseconds to see the tiger and turn and run the other way, I am much more likely to survive and be successful than if I look at the tiger and count the stripes and spend time trying to figure out, is this really a tiger, or is it just some, way that the light is reflecting off the leaves, and then I'm dead. Yeah, so those kinds of observations suggest that it's a strong evolutionary pressure to be able to perform calculations that the mind is performing in the right length of time. You also see associated with dynamicism arguments along the lines that a lot of cognition actually needs to be explained in terms of interactions between the system of the mind and the environment. So you have these interesting cases where, if you take a bartender who's an expert at mixing drinks, and they can do things extremely quickly in their usual environment, but then they'll change the weight of the glasses, or you move them around to slightly different places, to become much less able to be successful at mixing drinks quickly. And this is because presumably they are very tightly coupled to the environment. They're making assumptions about how the environment is going to behave, what the dynamics of that environment are, where things are. And so a lot of the cognition, as people like to say, kind of leaks out into the world. So there are aspects of what make us successful as reasoners and thinkers that are really external to us, and we are very strongly linked to that externality. And so dynamicists again want to say that this is another way of arguing for the importance of time and sort of understanding what happens over the barrier of our skin, right, when coming to explain minds and mental machinery. So these three views, of course, I just very quickly introduced them, but they have in a lot of ways set up arguments that are ongoing today. If you go to the Cognitive Science Conference, as I tend to go to every year, you'll see people really embracing one of these three views, arguing about why it's superior, or people will argue why other views have particular kinds of failings that they'll never be able to get over, or you'll have people trying to unify the views and say, oh, well, you've got the best of both worlds, and on and on. So I think, yeah, it's sort of a useful background to notice that really what we're talking about here, although we're going to dive into brains and talk about neurons and spikes and everything, down the road, you'll see after a couple of lectures that we seem to be adopting and trying to explain and touch on issues that are relevant for all of these different kinds of perspectives. And ultimately, I like to try to think that one of the things we do in the lab is essentially attempt to unify all of these approaches by explaining the same targets that they each have in mind, while taking an approach which is really grounded in the physics of biological systems. So with that in mind, I think it's also kind of obvious that we sort of have a greater affinity to connectionist type of techniques and approaches than to the other two, at least for our underlying methods, although ultimately, as I said, we definitely want to describe all of the phenomena that symbolicists and dynamicists really argue are critical. But because we have that affinity to connectionism, I think it's helpful to take a little detour into the history of connectionism. So as you may or may not know, original connectionists, by which I mean the folks who wrote the PTP books, didn't actually think of the networks that they were building as being equivalent to brains. They thought instead that they were brain-like. So they saw it as a kind of analogy, right? So just like the symbolicists had the mind as a computer analogy, the connectionists said, well, we think that minds are like these networks where we have a bunch of simple-ish nodes that compute simple functions. We connect a lot of them together. And if we connect them in the right kinds of ways, we get very interesting behaviors. And just to complete the analogies, if you look at dynamicists, they will say things like, the mind is like a dynamic system. Dynamical systems like the weather are ones we're familiar with that are very complicated and ultimately chaotic. And they want to think that those kinds of the kinds of techniques you use for describing those kinds of systems, differential equations and so on, are the right kinds of techniques for also describing minds. But back to connectionism and these brain-like networks, the idea wasn't originally that each of these nodes is actually a neuron. People would more often refer to them as nodes. Now people typically do refer to them as neurons or artificial neurons. They could stand for things like concepts. So one of these nodes could be a concept. One of these nodes could be an entire hypothesis, like a sentence. And ways of thinking about how minds reason would be to say, yes, some sentences have affinities for other sentences, which are governed by the strength of connections between those different sentences, and we could let these networks sort of do computation and settle to determine what sentences went together, and that would be a kind of reasoning, figuring out, what sort of solutions there were to particular kinds of constraint satisfaction problems, for instance. Or we could think of these things as concepts, right, like dogs and cats, or we could think of them as concepts and subconcepts, so like cat and has 4 legs and is furry and has a tail and so on. Not necessarily as neurons. Now, the approach that we take in most of what we'll be talking about, we actually do think of them as really being neurons. Right? We're trying to draw a fairly direct comparison between the elements in our models and neurons in brains and biological systems. But nevertheless, we actually do have a lot in common with these brain-like networks. So for instance, if we dive in a little bit and look at what's going on inside each of these nodes in standard connectionism, we'll see something that's actually pretty similar to what we'll see later on when we get sort of more into the biological version of this. So for example, we can write down this node here as being a function of its inputs, right? So we have input 1 and input 2, and each of those input nodes has a weight, W1 and W2, and ultimately the total input going into this particular node, right? So we could call this B1. So maybe this is this node up here. The input going into there is going to be a sum of A1, so the activity from neuron 1, sorry, that's a 1, plus A2, where each of those are multiplied by their respective weights, W1 and W2. So that's the sort of raw input, if you will, going into the node. And then you'll also notice this little step function here. And that is a nonlinearity of some kind. And that nonlinearity is actually extremely important. It's what a lot of people will argue, allows these to be computers in some interesting sense. And so ultimately, the overall activity of the node on the B side, so this receiving node, is going to be written something like this. It's basically summing the weights times the activities of the A neurons and put through some nonlinearity G. And if we want to be even more succinct, we can write this as a matrix multiplication, or where we have a vector of weights W and a vector of activities A, and that's going to result in the activity coming out of the neuron B. And all of these things are preserved in our more biological kind of approach, but of course we're going to add some things onto this. So one thing that you'll notice isn't here, for instance, is time, right? There's no notion of what happens if my inputs A are changing over time, or my weights, or what have you. Now, the big question, of course, that shows up even if you don't worry about time is exactly how do we pick these things, right? How do we pick these weights? And as many of you may know, the most common way to pick these weights is by using a technique called backpropagation. And backpropagation is really what allowed connectionism to have its heyday in the 70s and 80s and 90s. Actually, the heyday is ongoing, frankly. Before that, there were other rules for trying to pick these weights, but they were basically shown to be inappropriate or not useful for constructing functions of the kind of sophistication that people wanted. But with backpropagation, of course, we saw what is now known as deep learning come to the fore, and it has, by pretty much any measure, been extremely successful. Now, you've probably heard of all the cool things Google has done with backpropagation, learning Go, getting systems to learn chess basically from scratch just by playing themselves until they're really good at it. Those are using backpropagation in them. You also have all the image recognition networks, many of the networks that we sort of run into every day on our phones, where they're recognizing speech and all kinds of things, making recommendations to us about what movies we'd like to watch. Many of these things are using backpropagation, and it's been incredibly effective. It's let us scale up to extremely large networks. A lot of the biggest networks are ones people are using for natural language processing, like BERT and others. These huge networks are trained using backpropagation. Now, there are many challenges which still people worry about when looking at these networks. Here are a few of them. And one way of thinking about this set of challenges is kind of relating what you can do with backpropagation and artificial neural networks to sort of intuitive approaches like symbolicism, where we could, in symbolicism, we could literally read off what rules were being applied in order to make decisions. When you look at things like backpropagation networks, you have a lot of weights and nodes, and it's often very difficult to determine what different weights are doing, what each node is doing, and to turn around and explain to someone exactly why a particular decision was made. And this is becoming important in cases where people are making decisions about who gets credit or who doesn't get credit, right? So banks will have these networks they're running. They can put all the features of the person that they're talking to into the network, and the network will say yes, credit, or no, no credit. And then the person's like, well, just a minute, why did you deny credit to me? And it's not an obvious thing to come up with an answer for if all you have is a bunch of weights and nodes. Similarly, we can, when we have these big models like BERT, and they're clearly doing interesting language processing, we can try to look inside them and figure out how are they representing those concepts in language? How do they represent indexicals or articles or all the different parts of speech? Again, it can be extremely difficult to read that off of these kinds of networks. And sometimes we might say we don't care, but in other instances, if we're really trying to understand how people work, or if these models are sort of representing concepts in human-like ways, then it would be fantastic to be able to get at that kind of understanding of what these networks are doing. So this is one of the challenges, is basically understanding and being able to analyze exactly what the behaviors of these networks boils down to. And another one is one that I've already kind of raised, which is incorporating time. So I sort of gave dynamicist type arguments for why time is important for understanding the mind. But even if we just take a kind of machine learning perspective, we'll notice that often some of these deep learning networks struggle a little bit with time. So long time scales is something that people have been concerned by, because standard techniques like long short-term memories, LSTMs, don't do really well when they have a lot of time history to deal with in making a decision about what the next output that it should generate is. In fact, one of the reasons that models like BERT came along, which are transformer networks, is precisely because LSTMs weren't great at dealing with long histories. And so transformers are sort of feed-forward networks that give long history as direct input into the network. Incorporating time and dealing with time is not something that comes really naturally to these networks. As I mentioned before, those times are generally not real times, so there'll be time steps. But if we're trying to explain temporal behavior in biological systems, we want to know exactly what those timescales are supposed to be. Similarly, backpropagation usually avoids things like online learning. We can think of this as also related to time, because basically, allow the system to go into the real world. It's working over time, and then it wants to learn over time as well. Backpropagation is usually used offline, where you get a bunch of data, you train up your model to give those weights that we were looking for so desperately for connectionists, and then we deploy the model. And once we deploy it, it doesn't really change much. But of course, biological systems adapt all the time. They can learn from their mistakes. They can notice new patterns that they never saw before. And all this kind of learning is not naturally captured by backpropagation type techniques, but really important for biological systems. Terry, yeah?
Yeah, so just one thing that I like thinking about when thinking about the similarities and differences between deep learning and sort of the stuff that we're going to focus on more during this course. Certainly we're going to be using deep learning when it's appropriate, but all of these issues that Chris just brought up, they're all situations where if we know a little bit more about what the system needs to do, okay, we know that the system needs to incorporate time here, or we know some sort of idea of what the internal steps are that the algorithm needs to go through, we're going to be able to define parts of the model and find weights for parts of the model sort of automatically independently of the backpropagation. And that's gonna be a big part of what'll let us tackle problems that right now you can't do with just backpropagation. Because backpropagation is great if you have a bunch of inputs and you have a bunch of outputs and you have no idea what's going on in the middle. But if you do know some things about what's going on in the middle, then it's going to turn out that you can make use of that information.
That also reminds me that for the high-level reasoning perspective, one of the things that's often missing again in these backpropagation networks, this is related to online learning, in fact, is things like one-shot learning, which of course people are extremely good at, and transfer learning, where people have learned in one situation how to solve a problem, and then they come to a new problem and they can just very quickly, often through explicitly noting those similarities, i.e. using their high-level reasoning, bring all of that past history into this new context and use it immediately quite successfully. So again, that's another kind of online learning. And it's also maybe something we can get a better handle on when we, as you pointed out, try to break the model down into sort of smaller components, or components where we understand what's going on throughout the system instead of it being one big black box. So, right, so all of these challenges remain. This is by no means a way of saying that backpropagation isn't fantastic. We use it a lot. We will see it show up over and over again. We saw it in the first lecture. Yeah, it's more like we want to expand the toolbox that is available to people when constructing models in interesting ways. So this is one really important sort of current version of connectionism, right? It's kind of a software view. We're looking at the algorithms, figuring out what the best ways are to, pick those weights. Another sort of parallel thing that's been going on, a little bit more under the radar, I'd say, is the development of hardware that is good for running neural networks. And there definitely is hardware which looks sort of much more normal than what I'm going to be talking about in lots of ways. And by normal, I mean things like GPUs or special purpose chips, which are doing a lot of matrix multiplication, which is really important for doing deep learning, both inference and learning. But I think it's fun to look at neuromorphics as well, which has also taken a lot of sort of inspiration from noticing how important this kind of computation is, and that if you match your computer to the computation you want to perform, you can often get really impressive improvements in efficiency, for instance. Now, the reason I'm picking neuromorphics to focus on is also because they being neuromorphs, as they're often called, people building neuromorphic hardware, do care a lot about time in real time and generally communicate on their hardware in the way that brains communicate. So this is using spikes instead of activities. And so there's a much sort of closer relationship between this hardware and what we see in biological brains. It also means that they care a lot about issues like latency and online learning. And the reason that focusing on hardware in particular is of interest is because the sorts of advantages you get there are best realized in hardware. So, we can, of course, simulate spiking neural networks on GPUs and CPUs, which is perfectly fine for developing models and getting a good understanding of what kinds of compute you can do that way. But ultimately, if you want to build a real-world system, which is efficient and robust and real-time and all that kind of stuff, you want the hardware to do what the software is doing as well. I think at one point we did a calculation where we thought, okay, if we take Spawn and we run it on GPUs because they're convenient, off-the-shelf kind of hardware, and Spawn is only 6.5 million neurons. If we scale that up to the size of a full brain, which is like 80 billion neurons or something, then we will be using over a gigawatt of power using today's GPUs. And this is not something which strikes us as sustainable or desirable or possible, yeah. Frankly.
For context, that's a couple nuclear power plants. So a couple nuclear power plants to simulate one brain, that just seems like a bad idea.
So this keeps us, very interested and in fact using this neuromorphic hardware all the time, precisely because we want to worry about large-scale modeling, building these great big systems. Loihi in November released a great big 100 million neuron system, which is super exciting because it lets us to begin to scale these models to levels that they haven't been before. And as well, if we even just take a small slice of that 100 million neuron system, it suggests that we can put it in the world, put it on mobile robots, integrated in all kinds of interesting places, and yeah, do efficient, scalable, robust, and dynamic time-varying AI. Time varying is important if you want to do things like video and audio and control robots and all those sorts of things. So I think these two perspectives are quite interesting. That is the hardware perspective, which we're using neuromorphics to exemplify, and the software perspective, both related quite clearly to connectionism, but also both very modern, lots of ongoing research, and areas that we're deeply involved in and think are really exciting.
Sorry. I also find it fascinating that in order to really take advantage of the energy efficiency on this sort of hardware, you have to do everything in neurons. And so a lot of times you, even if it's a little bit of your algorithm, you're like, oh, I would just normally do this on a normal computer. The fact that you sometimes just don't even have that option, you have to rethink a lot of how you program. So that's, to me, that's what these things are forcing me to do, is if I really have to take seriously the idea of neural networks and that I do everything with neural networks, that changes how I think about programming. I can't do for loops anymore. And I'm really liking that exciting change, especially since if you can get your mind over that jump, if you can make that adjustment, you can make huge, you can take advantage of a lot of energy efficiency.
Yeah, absolutely. And even simple things like max functions, which of course were all over connectionist networks for a long time, are not necessarily super easy to implement if really all you have are a bunch of spiking neurons. And yeah, that actually brings us to my next point, which is that while the sort of historical roots of the kind of approach we'll be talking about today can definitely be drawn to connectionism, and we definitely use techniques like backpropagation, and we also definitely think that worrying about the hardware and being able to program this kind of neuromorphic hardware is critical and important. Ultimately, we started building these techniques looking at biological brains. As I mentioned in the last lecture, we take a little bit of inspiration from Richard Feynman here, where we're trying to essentially build brains which are sufficiently like biology that we feel like it improves our understanding of those systems. One way of talking about this is it's kind of a synthetic approach to neuroscience. While of course the sort of standard approach is analytic, meaning we gather a whole bunch of data, we look in the data for patterns, we try to figure out what those patterns are and explain them. We also think taking a synthetic approach can be helpful, where we sort of say, okay, this is our basic understanding. This is how we think neurons work. This captures like core elements of what neurons are doing. And we want to figure out how we can put a lot of those together in some way related to what biology is doing to exhibit interesting and useful functions. And so SPAWN is one example of trying to do that kind of thing. And when I say more synthetic, of course, people have been doing this sort of thing since the 20s, at least. But we want to sort of push in the direction of making these much, much bigger. So, trying to have networks which actually address cognitive behaviors and aren't just sort of stuck on the kinds of things that insects or rodents or something can do. And we also want them to be kind of complete in some interesting way. So I sort of feel like we can make a stronger argument that we have an understanding of how the system is working if we give exactly the same inputs to the model as we give to the biological system that we're comparing it to. So it's really nice to have inputs which are identical, and then input processing, which we hope reflects what's going on in the biological system, and then outputs, which again we can record the same from the biological system that we do from our model, so we can see behaviors typically, muscle movements and so on. And then, as I mentioned in the last lecture, you can of course dive in and get all kinds of data that is actually not accessible to you in the biological side. But overall, the techniques that we're going to be talking about for the rest of the lecture are starting at this perspective where we're like, what? Connectionism is cool. Symbolicism actually has a lot of interesting problems to solve, and there's a lot of super important cognitive behaviors that have been identified by symbolic models and modeled that way. Dynamicists have some really interesting insights in saying that, yeah, you can't leave time out of the picture, right? Ultimately, what you're building is a chaotic dynamical system. How do we integrate all of that stuff together? What's a unifying principle? And one place to start is to say, well, look, brains are physical systems. Physical systems have particular kinds of dynamics, and those dynamics are governed by the physical structure in that system. Let's see if we can try to start there and build up and get to the places that connectionists get with their, large-scale deep learning models, and get to the places that symbolicists get with their high-level cognitive models. So when you take this kind of approach, of course, you immediately run into some challenges, right? So these challenges include, well, when you look at neurons in the brain, they tend to be really different. So neurons in the cerebellum look very different than neurons in cortex, and neurons in basal ganglia have very different physiology than neurons in, pick your favorite area, thalamus. So, the heterogeneity that we run into means we can't always just borrow the techniques that connectionists often used, where they assumed every node's identical, right? They all have, the same, or maybe one of a small handful of transfer functions, input-output functions. Instead, we have to deal with neurons, some of which are, micrometers in size, and others of which are meters long, right? So I always like to ask people, where do you think you find a meter-long neuron? And you might guess it would be in a very large animal, maybe something like a whale or a giraffe. So there's like a 2-meter long neuron that goes from the top of from the giraffe's brain all the way down to its back leg, running down its spine. So yeah, neurons come in huge numbers of sizes. They also use a lot of different ways of communicating. So although in our models we often just have a weight, and we've got some output from a neuron and it goes through a weight into an input to another neuron, in real biology you actually have neurotransmitters happening in between there, and different neurotransmitters can cause different kinds of effects in the receiving neuron. So some neurotransmitters have really long, slow effects, things like NMDA, and others have really fast effects like AMPA. Both of these are excitatory neurons, so they will sort of increase the activity of a receiving cell, but they have very different time constants. You also have neurotransmitters which are inhibitory, like GABA. You have different kinds of GABA, like GABA A and GABA B, and these have different kinds of dynamics. And you have things like dopamine and serotonin, and these are other and the list goes on and on and on. So there's just a huge amount of complexity and heterogeneity, and when you're constructing a theory of how these things can be used to do computation and build up very interesting physical systems of the kind that we find in biology, you have to do something to account for that kind of heterogeneity, right? You don't want to assume everything looks the same. Similarly, we have a lot of noise issues in the biological brain. Maybe they're not issues, maybe they're actually something we can take advantage of. But pretty much any way that you look at it, brains are noisy places. So, one commonly cited sort of fact is that if you look at the likelihood that a neurotransmitter actually goes from neuron A to neuron B, can vary wildly from 90% likelihood that when you sort of have an action potential from the output neuron, that action potential actually turns into neurotransmitter and gets to the receiving neuron. That might be 90% of the time, or it might be 10% of the time, which means like it barely ever happens. So you have the first neuron saying, hey, I've got something important to say, and the second neuron doesn't even hear the message. So that kind of variability is well measured. You also have just the mere fact that you are in a warm brain. You have a bunch of Brownian noise. So there's all this variability which is coming from the mere fact that you're working in a biological chemical soup which is at room temperature. Actually, hopefully it's higher than room temperature. So it should be like 34 degrees or whatever it is, I don't remember. But it's warmer, right? Which means, in fact, it's even more noise than what we might expect otherwise. And yeah, you can do interesting measurements where if you see like exactly how long does it take for an action potential to go down an axon, right? So suppose I have a piece of axon, right, which is like the wire that's sort of coming out of a neuron going to the next neuron, and I start an action potential at time t. How long does it take to get to the end of the axon? And if you perform that experiment over and over and over again, you find that there's variability in that just mere transmission of voltage over what we would think of as a wire. The variability is small. It's on the order of nanoseconds, but it's noise nonetheless, right? It means that you can't assume that when you send a action potential at time t, it will be getting to its receiving neuron at exactly time t+1. It could be time t+1 and a bit, might be time t+1 minus a little bit, you just don't know. So lots of sources of noise, lots of issues with reliability. You also have more nonlinearities than people looking at artificial neural networks are used to. So as I mentioned, in standard neural networks, you've got these functions, which we drew an example step function. That's a nonlinearity. That's a nonlinearity that's occurring kind of over space. where you have your input here and your output here. But in biological neurons, you also have nonlinearities happening over time, right? So this is X. But in a neural network, you also have T, where you'll have nothing coming out of the neuron, and then you get a spike, and then you have nothing coming out of the neuron. So you have another class of nonlinearity that people haven't thought quite as much about, because, of course, as I mentioned, neural networks don't really model time per se. In fact, I think it's interesting. Let's be explicit about this. So a standard neural network, right? You'll have AI, that's going to be the activity of your neuron, is going to be some nonlinear function G of some input X, right? So that's what we would typically write down, as I did on the previous slide. For spiking neural networks, biological networks, we really want to write something more like this, that the activity at some particular point in time is a function of the input X and time, right? You really have another dimension that you are worrying about when you're building neural networks that are using sort of biologically inspired or spiking neurons. And lastly, scaling. So, if you've introduced all of these additional complexities at the level of individual neurons, and now you want to make really big networks, you want to have millions and millions of them cooperating together, how are you going to approach that problem, right? What techniques and methods and so on can actually get you from single neurons to millions of neurons? Now, unsurprisingly, we're going to have some suggestions for that. That will actually come a little bit later on. And right now, we're going to focus a little bit more on the single neuron level. Let's just start with single neurons and how can we understand what is going on inside single neurons. So what are these neurons good at computing? So unsurprisingly, they're good at time-based computation. As I mentioned, they really are best written as a function of both the input and time. They also tend to be fairly low precision. What do I mean by that? Well, people have done lots of experiments where they basically inject a white noise signal into a neuron. So you've got some sort of time varying noise, and you can inject this right into the neuron, and then you can record a bunch of spikes, right, coming out of the neuron, some interesting spike pattern. So this is time, and I'm just going to inject this like right into the cell. And then looking at this output, we can say, how well can we reconstruct that original input? And when they do that, they find that they can do a pretty good job of reconstruction, and the amount of error that is left over lets them know exactly how many bits per spike were available in the spike train. And it turns out that you get about 1 to 3 bits per spike coming out in biological systems a lot of the time. If it turns out to be not a white noise signal, but a sort of environmentally relevant signal. So the example I know is for bullfrogs. Basically, you have a bullfrog sacculus, which is like an ear where they can hear vibrations. And if you show white noise, you get like 1 to 3 bits. But if you show other bullfrog sounds, then this actually interestingly goes up to about 7 bits per spike. Okay, but it is still not super high precision. We're not dealing with 32 bits per packet, right, per piece of information as we are with digital computers a lot of the time. So it's probably reasonable to call this low precision. And part of the reason it's going to have to be low precision is because of all the sources of noise that I talked about last time. And then last but not least, we'll also notice that biological systems tend to be adaptive, right? So this is that online learning thing that I was talking about before, where it's never really the case that biological systems stop learning. We don't have an offline and online distinction in any sort of deep and meaningful way. So again, we're going to have to worry about learning from day one and also learning while the system is functioning, right? We don't want to have to stop it. We can't. And it's going to be functioning and be sort of working online all the time. And then we still need to update it and adapt it and not break it at the same time. What else are they good at computing? Sort of more generally, if we have particular kinds of nonlinearities, that's going to actually make us be able to compute particular kinds of nonlinear functions. We'll dive into this a lot more detail, not as much as we could. So in the NEF book, we actually go into this. There's an entire chapter devoted to what kinds of nonlinear functions you can compute well with neurons. And it's in some ways interesting that we can give a fairly compact mathematical specification of that. But one thing it does make clear is that neurons are going to be better at computing some functions than others. As Terry was mentioning, it's nice to be forced to basically only use the kinds of nonlinear functions that neurons are good at and not introduce ones that computers happen to be good at. And that's critical when you're using things like neuromorphic hardware. And then we run into this question, right? How much complexity do we actually need? Real neurons, we tend to draw them kind of like what we've got down here, right? We've got this nice long axon, there's nothing around, and oh, there's another neuron over here, and we started with this neuron over here. Look at all that space, isn't everything nicely laid out? Well, the real system actually looks like that. Things are ridiculously packed into tiny little spaces. All these neurons are connecting with each other more than once. The dendrites are near each other, so the dendrites are near dendrites, and sometimes they connect, and you get things called gap junctions. And if you look at any one of these individual cells, they have tons and tons of ions flowing through them of many different varieties. And there's active processes in each of the dendrites and axons, and you can basically use, and people do this, use supercomputers to simulate single neurons. They're so complicated. So can we build a model like Spawn with neurons that need supercomputers to simulate them? The answer is definitely we can't, right? So we have to make some kind of decisions about where we want to put our complexity. And so for our purposes, in the lectures here, we're going to be assuming a fairly simple kind of neuron. It's a neuron, it's called a leaky integrate and fire neuron, and you'll see it and we'll have chances to play with it in Nengo. So I'm not going to explain it into too much detail, but the important things about it are that it captures the neural spike. So you do have this, temporal component that's really important. It also has these sorts of nonlinearities that we showed before, like a step function. It's not a step function, but it's some kind of nonlinearity. It does model sort of voltages flowing in cells and through cells. It also models dendrites, and these sort of, you can see on here, these little arrows, these are currents flowing from dendrites to the soma. It models those as well. So we add on a little synaptic model that's going to capture that kind of behavior. And so you can see that it's actually getting a lot of what is almost always mentioned when people talk about neurons. Because we have a synapse model, we'll have something that captures the effects of these neurotransmitters. Of course, we'll have connections between them, which are going to have our weights in them. So, the networks that we build will have a lot of interesting biological properties. In fact, properties we can map onto real biological systems. So we will have time constants that are there because of the biolipid membrane in the cell, right, has that particular kind of time constant. But you can crank up the complexity a lot as well. And the NEF lets you do that, but for explanatory purposes and as a place to start, and also when you want to scale, sticking with the leaky integrate and fire neuron is often quite helpful.
I just wanted to also point out in there that all of the techniques that we're talking about should be able to work with, as you increase the complexity of the neuron, that's going to be fine. Specifically, what we're trying to do is we don't want to be limiting ourselves to only this particular neuron model. That turned out to be incredibly important when we got into the neuromorphic hardware, because the neuromorphic hardware doesn't exactly implement any, this sort of standard neuron model. And so it was really helpful to have made sure that all of our techniques are flexible in this way. That we don't have to we don't even have to decide to use spikes. If we don't want to use spikes, fine, don't use spikes. It's just another neuron model.
Yeah, that's exactly right. So yeah, so we'll often, in virtue of this, just write GI, right, and then some current, and this thing stands in for whatever neuron model you want to stick in there. So yeah, that will probably show up again. And we definitely are still doing work and research on making that GI arbitrarily complicated. So, it's not easy when you start making your neuron extremely weird, having all kinds of crazy dynamics, but we're working on it, and we've had more success than I might have initially suspected we would. But I also wouldn't want to suggest that we've kind of solved, being able to wire up any kind of neuron model you could possibly imagine. And I think the other important aspect here is answering this question, like how much complexity do we need? It really depends on what we're trying to do, right? So it really comes down to however much is useful for the question we're asking. So because we've kind of formulated the question that we're focusing on here as one of scaling, like we want to start from small neurons and get to really big models, then the leaky integrate and fire neuron is a pretty good one because it captures a lot of the biological elements that are preserved across many neural models, but at the same time is computationally cheap. And for that reason, we can use our supercomputing to simulate millions of neurons instead of trying to use it to simulate one. Now, if we begin to ask questions about, well, what happens if I introduce a drug and it manipulates, this particular neurotransmitter, as we saw in the previous lecture, we can do that. We can go into Spawn, we can say, okay, these neurons are going to be really complicated. We want, 15 or 20 equations per cell because we want to pretend that we're going to manipulate them with a drug. And if you need to do that, then great. You should definitely introduce that kind of complexity into your model to answer that kind of question. So I think there's no right answer to what the right amount of complexity is. It's really dependent on what your purpose is in building your model. All right, so this kind of has set us up for saying, all right, we've seen one way of setting all these connection weights. You've pointed out that, yeah, artificial neural networks are missing some sort of critical elements of biological neurons. Introducing those elements actually might be important for things like, advanced neuromorphics and AI when we're using sort of these new kinds of computational platforms. They are presumably important if we really want to understand how biological systems can scale up to cognitive ones. How can we bring all of this stuff together and actually figure out, what are the right ways for figuring out connection weights if we have neurons that act over time and so on and so forth, and try to worry about heterogeneity and noise and et cetera, et cetera. So that's where these methods called the neural engineering framework come in. So Charlie Anderson and I proposed these methods starting in about 1999 and maybe even a little bit earlier than that, and sort of working through techniques which really tried to take a biological approach to building computation. And at the time, we weren't explicitly worried about cognitive systems. We were more worried about even small circuits. How can we have a principled approach to taking what we know about, is a reasonable model of a biological neuron, something simple like the leak integrate fire neuron, but get it to do something interesting, something we see in biological systems. And really, it was a way of essentially letting people describe the function they wanted to get out of these neural networks at a high level. What is that high level language? Well, it's not Python. In fact, we thought of it at the time as dynamical systems or control systems. But once you specify the function you want, then can you get neurons to do that function? Can you build a multi-layer spiking neural network that realizes that function? And maybe it has to be recurrent, and maybe it doesn't. It's really going to depend on what functions we're asking about. So we formulated this framework in terms of 3 principles to try to make it clear. These are our assumptions. This is how we're approaching the problem. And what we're going to do now over the rest of this lecture is basically introduce the first of those principles. So principle number 1 is about representation. Principle number 2 is about computing. So how do you use the representations and compute with them? And then principle 3 is more focused on dynamics. And so that will be a separate lecture. So representation, computation, and dynamics. You'll notice that that is the subtitle of this book called Neural Engineering. Okay, so I'm going to start by just giving a quick verbal introduction to each of these principles, and then we're really going to get into the nitty-gritty, and then Terry's going to take you through running some examples which realize those principles in Nengo. Hooray! So principle number 1, representation. So in short, neural spike trains, so those things that we've been introduced to, these are generated by neurons in the brain. We think of those as some kind of nonlinear encoding of a vector space, and we think that that vector space can be linearly decoded. So what do we mean by a vector space? Well, that's really just an input of some kind, right? So a vector space could be an object moving around in the environment. It's XY position, right? That's a vector, 2-dimensional vector space. And we think that neurons are active, some neurons at least in the brain are active by mapping from that 2-dimensional vector space in some nonlinear way into the spiking activity of the neurons. That's not really surprising, right? People have thought that kind of forever. We're also thinking that you can typically linearly decode the original vector space from that nonlinear encoding. So if we look at the spike trains of a bunch of neurons that are encoding that space, and we just do a linear operation or a couple of linear operations of some kind, that will actually give us a pretty good estimate of what the original input was. And the reason we call this a representational principle is precisely because representation seems like something that you are trying to reproduce what the original signal was, right? So if you think of Morse code, for instance, you're representing the letters of the alphabet, A, B, C, and so on, in dashes and dots. And if you can go from those dashes and dots back to the alphabet, then you've decoded it and you've sort of preserved the information, i.e. you've represented those original characters. And we think the same thing's going on here, where we have some input vector space, some spike train, which is like the dashes and dots, and then we want to see, can we get from that spike train back to that vector space? And if we can do that really well, i.e. with no errors, we'll say we've got perfect representation. And if we do it with a little bit of error, we have good representation. And we know we'll be able to quantify exactly how much information was transmitted by that encoding. Now, representations are all well and good, and they're definitely a useful way of thinking about what biological systems are often involved in, but they aren't what those biological systems are doing ultimately. Really, the systems are transforming their inputs in interesting ways, or you might say computing over those inputs. So while we might know where a particular fly is at an XY location in the environment, what we really want to do is figure out how far is that from my arm. So I'm going to have to do some transformation into my arm coordinates in order for me to figure out how to move my arm to grab the fly. So doing these kinds of computations mean that we don't necessarily want to just reproduce what was stuck in, but rather we want to compute a function of the vector space that was stuck in. Nevertheless, the NEF is suggesting that we can do linear decodings of those inputs to compute functions of those inputs, right? So for example, I've got my XY vector space, I stick it in, I get a bunch of neural spikes, and then I weight those spikes in some different kind of way, right? So this is the alternate linear decoding. And in so doing, I can compute whatever function I want of that original vector space. Now, when I say whatever function I want, what I should maybe say is we can try to compute arbitrary vector functions on that vector space. And we can do a good job and a bad job, just like we did a good and bad job of representation. But again, if we take this approach, we can quantify how good a job or bad a job we can do. And that's the kind of thing that lets us figure out what's easy to do with neurons and what's not so easy to do with neurons. And the third principle, which as I mentioned, we're not really going to dive into today, in some ways really brings the other two principles together. So we call this the dynamics principle. And occasionally I could be persuaded that really this is the one principle of the NEF because it kind of has the other two principles as part of it in some important way. In this instance, we think that these vector spaces, which are being represented by spike trains, are something that we can define a transformation over in a sort of standard differential equation kind of way. So we say, this is the current value of my vector space, and here's the change in my vector space at every moment in time. And if we specify that, we can actually, write down every dynamical system there is, which is probably all the things that we really ultimately care about, because it covers nonlinear systems, chaotic systems, linear systems, of course, and so on. So sort of succinctly put, we would say that the neural representations from principle number one, so those vector spaces that are being represented by spike trains, we can think of those as state variables. We can call them control theoretic state variables like we did in the book, where we were thinking of things in terms of control theory, or we can just call them dynamical system state variables. And those state variables are basically defined by some, or they are the state variables of some nonlinear dynamical system. And a nonlinear dynamical system means that we're going to want to write down, how does my vector change as some function of itself and inputs in time, right? And this function f means that we need to perform a computation, and that's exactly what principle 2 is going to do, right? It's going to tell us how can we perform that computation using these alternate kinds of decodings. And if we take this to be the representation and this to be the function, then principles 1 and 2 come together and let us define any nonlinear dynamical system. So that's what we'll get to over the next 2 lectures. But let's, as they say, start at the beginning. So principle 1. Here's a cool movie of a goldfish eye. And you can see that what's being plotted at the bottom there is the spike rate. Hopefully you can hear the spikes. They're these little clicks, kind of like Morse code, which are changing their frequency. And the relationship between the eye position and the frequency of spikes coming out of this neuron is being plotted for you in real time. And you'll notice that most of them lie on that line, right? So in some ways, this line is telling us what is the nonlinear representation or the nonlinear relationship between the variable we're representing, the state space, i.e. the eye position, and a bunch of spikes coming out of this system. super cool movie. All right, so this is an example of an encoding, right? We've got input, I position. We've got a nonlinearity. You can see that, for part of this, there's no activity here, right? So there's a nonlinearity right there. And then we can hear the temporal nonlinearity, which is the spikes. Okay, so that's an example of an encoding. And then the question is, well, what were we going to do to represent the input? Well, we were going to decode it in some kind of way. Here's an example that you can sort of grasp because it's simple enough. We're just looking at two neurons, and we'll call them Alice and Bob. And these two neurons are going to be encoding some kind of input, something like I position or whatever you want, any variable X. And they're going to be communicating to Charlie. And essentially what we're going to be asking ourselves is, what could Charlie know about the input that was going into Bob and Alice, right? So to be clear, we've got this variable X, and that same variable is going to be driving both Bob and Alice. Now, Bob and Alice are neurons. They're neurons. They're projecting their axons to Charlie's dendrites. So Charlie has connections to both Bob and Alice. And when that input is going into those neurons, it's generating these patterns of spikes, and they're going to be, slightly different patterns of spikes for different neurons. Those flow down the axon to the synapse, where they cause neurotransmitter to be released. The neurotransmitter flows across the synaptic cleft and causes a current in the dendrites of Charlie, and those currents then flow into Charlie, and that's basically what Charlie as a cell sees coming from Alice and Bob. And then, of course, the process continues, and Charlie will stick out some pattern of spikes to communicate to who's ever downstream from Charlie. All right, so we can begin by, taking our perspective of Bob and Alice as sort of having these tuning curves. So for instance, we might say, okay, we're going to have this variable X being injected into Bob and Alice, and maybe Bob's tuning looks something like this, right? So this is reminiscent of what we just saw in the I position system in the goldfish, where we've got some variable here and some nonlinear function, which is also drawn in red, that is telling us what kind of firing rate we expect to come out of Bob as a function of the input X. And we can do exactly the same thing for Alice. And here we've got Bob and Alice having slightly different tuning curves, which is what you typically find in the brain. You have a little bit of heterogeneity there. But the question for Charlie is, well, how could Charlie infer what X is? Now, the reason we're asking this question is a theoretical one, right? We want to know if we could reconstruct X from Charlie's perspective, we could say how much information Charlie has about that original input variable X. This doesn't mean that we think brains are doing this kind of decoding and that each neuron is asking the question, oh, what was the input to my preceding neurons? But instead, it gives us a theoretical tool for understanding information transmission through a neural network. Now, clearly, we haven't introduced spikes yet, right? We've only got one of the two nonlinearities captured there. So let's think about the other one, right? This is nonlinearity over time. So now I am drawing a input signal, which is varying over time. And we can presume that we have both Alice and Bob again, right? So we'll have the same two neurons that we had before. So they're getting the same input. And in virtue of the fact that they have slightly different tuning, they'll give different kinds of spiking output. So for instance, Alice might give spikes where for large values of X, spikes start coming out. And when values of X are low, there are no spikes. And Bob, in contrast, because you'll notice that Bob was sort of tuned in the opposite direction to Alice, might give lots of spikes when the signal goes low or negative, and no spikes when the signal is high or positive. Now we have to ask ourselves this question about what could we do to reconstruct that input? And a linear decoding, which is of course what the NEF was assuming, means that maybe we can think of those postsynaptic currents that are generated in Charlie as essentially spreading out the information from these spikes over time, and we could just sum up those postsynaptic currents, and that will give us some kind of estimate at least of what the original input is. Now that sort of makes sense until we notice that, well, Bob actually is giving spikes when the signal goes down. So really what we're going to want to do is multiply Bob's postsynaptic currents by -1, right? So now we've basically got two elements to our linear decoding. We've got a weight, right? So we've got +1 here for our weight, and we've got -1 here for our weight. And then we've got our sort of temporal linear decoding, which is this postsynaptic current shape, which is the same in this instance for both Bob and Alice. So if we do these two kinds of linear decoding, one over time and one over space at the same time, and we just now add all of those things up, right? So all we've done here is taken these spikes, turned them into these postsynaptic currents, and then summed them with all the other spikes, and you get this kind of wiggly line. What you notice about that wiggly line is, hey, it's kind of going up and down in the same way as my input X. Big shock. Now it's not fantastic, though definitely there is error there, but there's also, clear correlation. And so it seems like definitely some information is getting through, and we can begin to characterize how much information is getting through, how much information per spike, how much is each of those neurons contributing to that representation, and so on. But in a nutshell, this is the representational principle of the NEF. That is, we have nonlinear encoding, which turns our input vector space, in this case it's just a scalar x, into spikes over time. And it's turned into spikes in this sort of spatially nonlinear way as well, in terms of the tuning curve. And then to get that information back out, we want to do this linear postsynaptic current decoding. Now I've stuck an extra word in here, optimal, right? So for this example, I just picked plus and minus 1 because that's super convenient, but really we can pick any weights we want, so we might as well pick the best possible weights. We could also, so that's sort of, optimizing the weights, the spatial decoding. We could also try to pick optimal postsynaptic currents to do decoding as well. But there we run into a bit more of a challenge because biology sort of defines postsynaptic currents for us. And if we calculate optimal postsynaptic currents, we find that they look kind of like, actually surprisingly maybe like real postsynaptic currents, but they're not exactly the same. So in the interest of biological plausibility and realism, we will use actual models of postsynaptic currents instead of making them optimal. But for the weights, that's something that we can definitely optimize for. So in Nengo, we can build these models pretty easily. We've got our Bob and Alice again here. And we've got the input X changing over time. We can see what the subthreshold activity of these neurons is. So this is the subthreshold voltage coming out of Bob and Alice. So if you stuck an electrode inside one of these neurons, this is what we'd expect to record. And then you can also look at the spikes, which are coming out of these neurons and being sent to Charlie down the road, as it were. And we can see that the activity of one of those neurons, this should look pretty familiar. The activity of one of those neurons is equal to some nonlinear function, right? So for us, that's the weak integrate and fire neuron, which is going to have as input x, so that's our state variable. And then we've got these two other parameters. So this one is what we think of as a gain. So as we change the gain, we would think of kind of like this slope of this neural response would change. And then we have this thing called the encoder. I haven't really talked about this yet, but it will definitely show up over and over again. It becomes much more relevant when we're using more than one dimension. In the case of one dimension, we can think of basically Alice as having an encoder of +1 and Bob having an encoder of -1. So we can think of those as on neurons and off neurons, or what have you, but you really do need to say something about the tuning of this particular cell to that variable x, because it can vary. It can be highly heterogeneous, in fact. And then this last one is what we call a bias current. And that is there as a matter of matching biological data to essentially shift these curves into different parts of the state space. And we can have long conversations about where the bias current comes from and so on. But altogether, these inputs basically determine what is the current J going into that neuron, right? That is then driving that cell to have the particular voltage that it has, right? This is the voltage that it has, and then also ultimately what the spike trains coming out of that neuron are. And then on Charlie's end of things, right, we can also write an equation down where Charlie now is trying to estimate what that original input variable x was. And what we're doing for Charlie, as we said before, is just summing everything up. So here, we can think of this as being, those filtered spikes, those postsynaptic currents. So every time there's a spike, we place the postsynaptic current. That's going to be these AIs. So that's really a function of time. And then these decoders, D, those are going to be those weights I was talking about, right? Where we just had plus and -1 on the previous slide. but we could also figure out what is the best weight we could have to give us the best estimate of x hat. And so we can also plot all of these things. So down in the bottom here, we've got the postsynaptic currents that would be generated in Charlie from Alice and Bob. Then we have the weighted version of those postsynaptic currents. So this is where we've weighted the postsynaptic current by the DI. So this is DI times the AIs, which we're calling the postsynaptic currents. And then lastly, here we're summing over all the I's, right? So this is summing over the I's and giving us the estimate that we have of our original input signal, which, like in the previous slide, is not really smooth. You can tell the input looks more like a sine wave, and the output, yeah, so much like a sine wave, but kind of like a sine wave, right? There's error there, but there's also structure. So this is kind of the simplest example that we can build in Nengo. And right now I've just been throwing this at you as a bunch of equations and pictures and movies, but it's time, Terry. Time to.
Yay, we can do hands-on examples.
Exactly.
I'm going to show a little bit quickly about how to just install it. So I am assuming by this stage that you already have Python installed on your computer. If you don't, then we recommend installing Google for something called Anaconda and install that. There's versions of it for every platform. It's basically Python with all the add-ons that you could ever want already pre-installed. Once you have that, if you go to your the command prompt or the Anaconda command prompt, in order to install Nengo, it is just pip install Nengo, and that will go and automatically download Nengo and install it for you. Pip is sort of a built-in Python automatic installation program. That gets you the core simulator, the core way of defining your models and running them. But as for all of these tutorials, what we're going to be doing is we're also be using this graphical interface. And so we're also going to separately install that. So pip install Nengo GUI or Nengo-GUI. That will get you the Nengo GUI. And once you have both of those things installed, in order to run Nengo, you will type Nengo. Perfect.
Yeah.
So what should now happen is your web browser should pop open, and you should get something that looks kind of like this. What we have is on the right of the side of the screen, we have just a normal text editor. Let me go up to full screen there. So on the right side of the screen, we just have a text editor. If you have your own favorite text editor, yes, you can go use your own text editor, but this one is built in. The file that is currently being edited is shown at the top of the screen here. And I'm just going to click on this and give it a new file name while I'm thinking about it. So we'll just call this example1. That file will be saved in whatever directory you were in when you started Nengo. So on the right side of the screen is your code. On the left side of the screen is a graphical display of the model as you're building it. Okay. So what do we want to do? What we want to do as our first example is let's start fresh with an empty file. If you are familiar with Python already, wonderful, that's great. This should be a little bit familiar, but the programming language we are using is Python. My hope is that even if you're not familiar with Python, it's at least somewhat clear about what the programming language is doing, and hopefully this can be a reasonable introduction to that. The beginning of any model that you build in Nengo is going to look pretty similar. Every time you're building one, the first thing you're pretty much always going to want to do is import Nengo. So that's basically a Python command that says, all right, we've done that pip install Nengo thing. Cool, now we have access to all of the components of Nengo. And anytime we want to refer to a component in Nengo, it would be something like Nengo dot something. And then the first thing we're going to do after that is we're going to create what we call our model. And the model is going to be a network. And the network, so the model is the thing I'm defining right now. Fine. And what a network is it's just sort of a container that just, it's just a bag to put a bunch of stuff in. So all of our inputs, all of our outputs, all of our neurons, all of our connections are all just going to get all as part of what we're going to call our network. Okay. So fine, just always use that as the first couple lines of your code. The next thing we're going to do is we're going to tell Python that all of the commands I'm going to specify next are all things that should get added to this model. Okay. So that's this with model command. So now we're just going to say, look, I'm going to give you a bunch of things, all of the things I'm about to create, put it inside this model. You will notice that there's a little bit of an indentation there. Python is one of these programming languages where indentation matters. So the editor automatically went and said, okay, I'm going to add some spaces there to indent the text. Just be aware of that and follow along with the example. All right, so we have an empty container that we can put a bunch of stuff in. For our first example, we're going to do exactly the thing that Chris just defined. We'll start with one neuron, and then we'll go up to 2 neurons, and then we'll go up much quickly past that. But let's start with one neuron. The way that we're going to create this neuron, neurons just generally don't ever exist totally on their own. They're usually in a group. So we're going to make this a group of 1 neuron. But instead of calling them groups, we call them ensembles. So I'm going to say that I have an ensemble. Basically, that's just a reference to the fact that it's not just a group of neurons, it's a group of neurons that are sort of working together to do something. And whenever we create an ensemble, one of the parameters that we always have to specify is the number of neurons in it. And there's going to be 1 neuron. And then the next thing we're always going to have to specify is, all right, the thing that this neuron is representing, what's the dimensionality of that space? Is this a 2-dimensional space, like an XY location? Is this 1 value, like in just a numerical value, just in the examples Chris was just showing? In our example, our input is just 1 value, so our input is 1-dimensional. So I have 1 neuron. It's representing 1 thing. Fine. I need to know about how this neuron is representing things. And in the diagrams that Chris was just showing, there was a couple sort of key things that were being specified. So one thing that was just always came up was this encoder thing that Chris said, this E variable. And that is just sort of saying, fine, this neuron's representing a 1-dimensional value. Does it fire more for +1 or -1? That's just sort of like the simplest aspect of this representation that we can have. We call these encoders. We're going to make this neuron fire more for +1. Okay. In general, that's going to be a list of multiple things. This is just a list with 1 thing in it, so that's going to be our syntax. The next thing we're going to specify is, fine, I want this neuron fires more for +1, but how fast does this neuron fire? Like, does this neuron fire at 10 Hertz? Does it fire at 1,000 Hertz? What's going on? All right. And so now we're going to say, well, this neuron's maximum firing rate is going to be, say, 100. Whoops, 100. And then the last thing we're going to specify is its intercept. So on that tuning curve plot that Chris was showing, there was some point along the x-axis where it goes from not firing to starting to fire. And so that's the x-intercept. And I'm going to say that starts firing at about -0.5. We'll get a graphical depiction of what that looks like in a moment. All right, so that's a bunch of stuff that we've specified. Basically, we've defined everything about our neuron. We could specify a lot more things. So like one other thing that we could also have specified is the neuron type. For now, we're just using the default, which is the leaky, integrate, and fire neuron, but I'll just include that for completeness' sake. But that now says everything about our neuron representation. So we're just saying, look, this is the neural representation that we would really like this neuron to have. Okay. Now, if I press play right now, so the bottom right of our screen, there's a little play icon here. Oh, actually, first, before I do that, I just want to note that when I was doing that typing, things appeared over here. So this is the pool of neurons. Our icon just has 6 circles, so the icon doesn't change based on the number of neurons. So this is a representation of 1 neuron. This graph that appeared, we're not going to use it yet, so I'm going to get you to right-click on that graph and remove it. But in general, this thing on the left is going to be a depiction of what's going on, and you can drag things around, you can resize them, and you can right-click on them in order to make particular plots. And the particular plot that I want to look at is I want to look at the spikes. Here we are. All right, so if I press play right now, what's going to happen? So I'm going to press play down here. And then so I can hit play, and then I can hit pause again to stop the simulation. What are we seeing here? All right, this graph, well, first of all, this graph is a little bit small. Let's make it a little bigger. There we go. We're running our simulation over time. The units on here are seconds. So this neuron is sitting here spiking. You'll notice I haven't given it any inputs yet. And that's because this is exactly what we've asked this neuron to have. This is sort of an implication of what we've asked the neuron to do. If I right-click on A right now and go to details and go to plots, we get this sort of tuning curve plot. This is if you held the input constant to this neuron for a long period of time, what would its average firing rate be? This is sort of a summary of the behavior of the neuron. And this is exactly what we asked it for. We asked it to be more sensitive to the input 1, so to fire more for positive numbers, fire less for negative numbers. We asked it to have an intercept of -0.5, so that's the point where it starts firing. And we asked it to have a maximum firing rate of 100 Hertz. So that's where all those parameters came from. But you'll notice that means that if our input is 0, it should be firing at some amount, just over 40 Hertz, 50 Hertz, right? And that's exactly what we're seeing when I'm giving it no input whatsoever, and I'm getting some spiking coming out. So that's spiking behavior.
But Terry, why is it spiking if there's no input?
So
neurons have to have currents get injected in them in order to spike?
Neurons definitely do. And so this becomes a question of what level of biological detail do you want to have? In the definition of the model that Chris was just showing, if there is some sort of bias current, if there's some sort of background current that is just always flowing into these neurons, regardless of what's going on with the normal inputs, that would be responsible for this sort of spiking. If we want to go ahead and model that, there's a lovely add-on to Nengo that's being worked on that adds in those sorts of low-level biological features. In this case right now, though, we are just imagining that there is sort of this background current being injected into these neurons, and that amount of current has actually been computed in order to make these maximum rates and intercepts work.
So yeah, I was asking the question partly just to also clarify that that graph we're looking at is not the current. It's not the input current, it's the state input. It's the input of X. And then there's some mapping, as you just pointed out, from that state onto the current. So even though it says 0 is equal to X, because of the bias, the actual input current to the neuron is some value higher than X, obviously.
Yeah, and sometimes that gets in the neuroscience literature, that can be the distinction between a tuning curve and a response curve, where the response curve is sort of, given a certain amount of input current, how fast or how much firing does the neuron do? Whereas these tuning curves tend to be more, given some sort of external stimulus, some sort of thing in the real world, something being represented, what's the firing rate of the neuron going to be? Okay, so we have so we have no meaningful input to our system, to our neuron right now. There is some sort of background input that the system has automatically put in order to give it, this neuron, the tuning curve that I've asked for. But there's been no meaningful input to the neuron yet, or no input that can change over time. So now we're going to introduce the next sort of component when building things in Nengo, and that's we're going to call a node. So we're going to call this just anything that's not a neuron is going to be a node, and the simplest node is just sort of an external stimulus that can go feed into this. So we're going to call this a stimulus. It's a Nengo node, and this node, in general, we can specify a function here that would sort of, here's some changing input that changes over time. I'm going to be just really simple and say, look, this is a node that just sends in the value 0 all the time. The user graphical interface will let me change that in the future, but I'll show how the graphical interface will let me change that. But for now, it's just a node that generates 0 all the time. And now I'm going to need to actually connect that stimulus. So first of all, you will notice the stimulus appeared here, a little box appeared. But now I actually need to connect the stimulus to this neuron. So this is going to introduce our fourth component, which is a connection from the stimulus to A. Okay. And now I have an input. So now we have a little line there, right? I can right click on the stimulus and, oh, well, okay. First of all, if I press play, all right, now I'm injecting 0 all the time. All right, my firing rate has changed. If I go ahead and say, all right, let's inject, so the firing rate was the same as what was before. If I say, all right, inject a 1 all the time, now that should change the firing rate. So now that the neuron should fire at 100 Hertz. Right, so we should see, well, oh, yes, the neuron is firing faster. We'd really like instead of like manually changing the code here, it'd be nice if we had some sort of user interface to do that, and we do. So if you right click on the stim object and click slider, then we get a slider that'll let me change that value on the fly. Now when I press play, I can go, ooh. Okay. And so this is just showing that we have got the same sort of encoding that we've asked for. Hey, when I input a negative number, the neuron does not spike. When I start getting into, when I get to about minus 0.5, that's when the neuron starts to spike. And then as I get for a larger and larger input, it will spike faster and faster. Okay, so this is just sort of defining one neuron with a particular set of tuning properties that I say, that I particularly want. Okay. No, actually, first thing I'm going to do is I'm going to save this. There's a little icon up here that says save. So we can click on that to save the file. I can also then, if I want to, since I'm going on to another example, I can click over here and say, all right, we're now going to change the file name, example 2. And now I'm going to change the number of neurons. So I have 2 neurons. If I say there's 2 neurons, I'm going to get an error. Right? It's going to say, hold on a second here. The encoders, there's something wrong with the encoders. Of course, there's something wrong with the encoders because I've said, all right, there's 2 neurons, but the encoders are 1. I now have 2 neurons. I'm going to need 2 encoders. So this is going to say, all right, the first neuron responds to, fires more for +1, and this other neuron fires more for -1. Okay. I have to similarly, I'm going to do the similar things for the maximum rates. We'll keep that the same, and the intercept we will also keep the same. We will see very, very shortly, certainly when we start wanting to get lots more than, many, many neurons, certainly you do not want to manually specify these things. We will get rid of that specification in a second. But for now, we're just being as explicit as possible. So now I have 2 neurons. Go ahead and run that. And now I have 2 neurons and they respond. So this is like with the Alice and Bob example that Chris was saying about, this is sort of a neuron that fires a lot for -1. That was Bob. If I go up here. This is a neuron that fires a lot for +1. That's Alice. You'll notice Bob does not fire anymore. And when we're in the middle, they both fire a little bit. And this is sort of relatively typical of what you would see in sort of biological systems. So all that's saying that if we can actually click here on these details and go back to the plots, and now we see here are the two tuning curves for those neurons.
Okay. Probably show the decoding as well by pulling up the
Sounds
other plots.
Good. So we've got these two neurons, and this common thing that Chris is saying is, can we recover what the input was given those two neurons? So if we take A, if we right click on A and we go to value, so we're getting a different plot here. This is going to be a plot where, so it's going to be the same data that's being shown in this spike, but we're going to do the two linear filters. We're going to do one linear filter that is the one over time, and that's saying every time there's a spike, that's actually going to cause the value to change and then gradually decay back. But then we're also going to do a weighted sum of these two things. Okay, so there's going to be some weighted sum. So in the example that Chris was showing, we take one of the neurons, multiply it by 1, the other neuron multiply by -1, we add it together. If we go ahead and do that, okay, then we get the plot over there in A. Now you will notice that is doing something like the input. All right, and that as it goes up, we get a value near the top. As it goes down, we get a value near there. Perhaps for comparison purposes, I can right click on stim. I will also pull up a value plot from the stimulus. So that's the actual stimulus. And I'm just going to overlay them on top of each other. And so now you can sort of see, there's that blue line in the middle. That might or may not be visible. We'll go put this down here. Now, since as Chris was saying, the thing that was optimized, we can optimally find what the weighting should be. But there's also that free aspect of, well, what biological, when there's a spike, how long does that neurotransmitter stick around? That's something that is often we're grabbing from the biology. So that's something that we want to be able to configure. So if I right click on A and I say set synapse, now I can sort of say, when I'm making that plot, what sort of synaptic time constant should I be assuming? Right? And the default is 10 milliseconds. That's sort of at the faster side of things you find in the brain. Let's go with the sort of the longer side of things you'll find in the brain, say 100 milliseconds. Right? So exactly the same model, same neurons, and now you will see that as long as that we get a much better estimate, now that we're taking that information and smoothing it over time, that those two neurons are doing a pretty good job of representing the input. Again, I'm going to try overlaying these.
I also like it when you show the bar, like the time bar, and how you can spread it out so you can see more history.
Oh, yes. So other sort of useful things to do in the Nengo interface, since you've got this running over time aspect, this bar across the bottom actually maintains a history of the past. And if you click on this blue bar at the bottom, you can drag it back and sort of see what you did in the past. All right. But then the other thing you can do is you can expand the blue bar, and that's going to say how much of the past should be plotted. So now, again, it's exactly the same data. We're just changing how it's being plotted. But then we can see just how well those two neurons are doing at representing that information.
Yeah, and it also makes the nice point that when you had the 10 millisecond filter on there, the error was higher. Essentially, you would say that you're losing information, but with the longer filter, you're also paying a bit of a price in responsiveness, right? So if you sit there and wiggle things fast, it's going to deviate in some interesting ways.
Yeah, and so you actually see that if I pause the simulation and then change the input and then unpause the simulation, you will see that it takes a while for that estimate, for that decoding value to go to its new level, right? And that's going to take longer. Whereas if I switch back to the faster time constant, synapse to, I don't know, whoops, wrong one. Synapse to back to 0.01, we'll go to 0.02. Okay, and now when I run that, if I pause it and change the input, now you'll see it's a noisier estimate of the value, but it tracks it better. So this is a very nice sort of trade-off that biology might have to do. Or, I mean, the other way that, as we will see, if we increase the number of neurons, we can also do much better than that.
That sounds like a perfect segue. Right, so as Terry was just mentioning, another way that we can improve the accuracy of the network is by adding more neurons. So, this is exactly the same kind of graph that we saw previously, where instead of 2 neurons, we now have, I think, on the order of 40 or 50. But we have all the same kind of parts where, right here, we're seeing all of the voltages, the subthreshold voltages. This is another graph that you can pull up in the Nengo GUI. We can see all of the spikes for each of the different neurons. This is definitely beginning to look a little more raster-like in some ways. Over here, we've got this activity view, where this is kind of like looking at a neural sheet, where we've laid out all the neurons in a spatial grid, and you can look at how active the different neurons are over different moments in time. You can see that the input is exactly the same as it was before, where it's just a sort of simple sine wave. And of course, nothing about the math is going to change. So we still have, for every neuron ranging from I = 1 to 50 is going to be described by this equation. But of course, we've picked sort of a bunch of these encoders, plus and minus 1. We've picked a bunch of different gains and biases in order to have heterogeneous tuning of the kind that we see over here in this picture. Then the decoding is also going to be exactly the same, where now we're finding a decoder for each of the neurons. We're filtering all of those spikes to give us the postsynaptic currents, which are going to look something like this. So this is all of the postsynaptic currents that are going into Charlie, assuming that he's still receiving all these 50 inputs. He's getting all of those currents. It's then weighting those currents to give something that looks like this, and then summing across all of the currents to get something that looks like this. And now that actually looks an awful lot like a sine wave, right? And this is even for a very short time constant. So this is still with about a 10 millisecond time constant. The other thing that we'll be looking at in this example is trying to understand where those decoders are coming from. Now, you might have noticed in the previous example, Terry sort of just ran the program and graphed stuff, and everything popped up and looked decoded in some useful way. So there's something going on behind the scenes, which is figuring out these decoders for us. But I think it's also pretty natural to think about learning those decoders. And so what we'll do, or Terry will do shortly, is basically introduce this rule as a way of learning those decoders, where we can say, oh, we're going to start with all the decoders equal to 0. So we'll just pretend all our decoders are nothing. And then we're going to run online over time. So as you might remember, I was sort of emphasizing the importance of online learning. We're going to run a rule, which is going to be this one, where we're going to have the error, which is some function of time. We're going to have the activity of the input population. So this is the presynaptic activity. And then we have some learning rate, as you typically do, just to determine sort of how quickly you're changing the rules, or the weights. And the combination of these is going to basically give us a method for updating all those DIs over time, such that we're decoding whatever we want to be, depending on this error. So basically, if that error is a representational error, it's saying, okay, we want to get back out the same thing that we had on the input side. We have this nice sine wave. Then we're going to use that, the difference between that and whatever my decoded estimate is, as the error. And doing so means that it's then going to find weights which are going to be minimizing that error, right? And this is quite nice in many ways. It's a fairly simple kind of rule. It probably looks familiar to many of you as a delta rule. We tend to call this the prescribed error sensitivity rule because when we originally derived it, we actually treat it as a kind of Hebbian rule where we have pre and post synaptic activity and it's all spiking. So, it looks a lot more like a sort of standard Hebbian rule with error. But, ultimately you can also think of it as a sort of delta rule when you're just thinking about these decoding weights. So I'll come back to that, thinking of it as sort of standard connection weights. Right now, we haven't really talked about connection weights. We're just talking about these decoders at this point. So with that, I'll turn it over to Terry, and he can take us through adding more neurons and then learning those decoders.
Sounds good. I will comment before you go there that the delta rule, so that rule that's written there is exactly also the first step in backprop. So that's backpropagation without the backpropagation, because there's only one set of weights. So we don't need to backpropagate the error to another set of weights. So in this particular case, you can also think of this as also being backprop. But yeah.
The very beginnings of backprop, at least. Yep. All right, over to Terry.
So first thing we're going to do is we need more neurons. First thing we're going to do is we're going to save our file, click on save up there, then we're going to go over here, and then we're going to save this as a new file name, example3. First thing we want to do is we want more neurons. Let's do 100 neurons. If I say 100 neurons, I really don't want to now specify a big, giant, long list of, I don't know, neuron 1 responds to +1 and neuron 2 has this firing rate. I don't want to specify those things. If I specify nothing at all, Nengo will randomly choose defaults for me. And the default in Nengo is to randomly vary the encoders around whatever space we're currently representing. Randomly vary the maximum firing rates to between 200 and 400 hertz. And randomly vary the intercepts between -1 and +1. Those are all uniform distributions. And so that is a great default for pretty much every situation. So a lot of times we can just get away with just don't specify anything. And indeed, since the leaking rate and firing neuron is also the default, we're even going to get rid of that. So just to start making our code look a little smaller and simpler. So that is now going to be our give me 100 neurons, and we're automatically going to put in a bunch of heterogeneity to get a variety of neuron tuning curves. All right, so that's going to simplify things a little bit. Now, so let's actually just run that just to make sure that's working. So here we go. We have a bunch of neurons. Let's show some of these plots. If I right-click on this spiking plot and say set number of neurons, I can show all 100 neurons. Just by default, it only shows 10 of them. And so now as I vary my input, I get different spiking activities. Okay. I can also show, just for completeness' sake, that voltage graph that Chris was pointing out. So this is sort of what's that internal membrane voltage of some of these neurons. So that's this plot here. So those 5 lines, those are 5 different neurons. And again, you can sort of see how their internal voltage changes over time. All right, well, so we're only showing 10 of the neurons, fine. If you right click on the graph, you can change the number of neurons, but I'll leave it like this for now. These graphs here are being it's just that Nengo is automatically computing the best decoder weights, the optimal decoder weights. That's why there's a bit of a pause when you press play. The gear spins a little bit. That's Nengo doing that optimization in order to find those best weights. What if we want to try to have that learn on the fly? What if we want to do online learning to have the system learn what those weights are? So first thing we're going to do is define, we're going to create a new node that is going to represent our output. Okay. And this is going to be a node that, so in general, it's like this node that we defined above, that had no input and just one output. This is going to be a node that I just want it to do nothing at all. I just want it to collect together its input and just show what that input is. So I don't want this node to do anything. So I'm going to say none, but I do have to tell Nengo, hey, is this node representing a one-dimensional thing or a two-dimensional thing or a three-dimensional thing? And so I do have to say that the size in is 1. Okay. And this node, these nodes that don't do anything at all are sort of shown graphically in the Nengo interface just as a dot, just as a reminder that they're literally doing nothing. Which is sort of convenient for us to be able to plot things. Now, the reason that I wanted to do that is I'm going to make a connection from A to that output, and that connection is going to be the set of weights that we're going to learn. All right, because I've got 100 things, I've got 100 neurons in A, and I've got one component here, output. So that means I'm going to need a 1 by 100 weight matrix in the middle here in order to take all those values and turn them down into 1. Okay. So let's go ahead and create that. So this is going to be a connection. This is a connection that we're at the moment we're like thinking about things right down at the neuron level. So I end up having to say I'm going to make this a connection from the A neurons. When we introduce sort of other ways in Nengo of making connections that don't involve manually specifying connection weights, we'll get rid of that dot neurons. But for this particular case, what we're doing is we're saying, hey, I want to think of A as just a group of neurons, and I just want to learn these connection weights. So I'm going to make a connection from A to output. If I just do that, Nengo's going to yell at me a little bit. Again, we get an error message down here saying, I'm expecting something that is a 1 by 100 matrix. Oh, yes, that's because A.neurons is 100 things. The output is one thing. I need to just specify a weight matrix. The way you specify a weight matrix when you're making a connection in Nengo is you say transform, and then you specify the weight matrix. And the weight matrix that I want here is just a bunch of zeros. Right? The easiest way to define a bunch of zeros in Python that is a 1 by 100 matrix is to use the NumPy library. So we're going to go up to the top of our code and we're going to say import NumPy as NP. NumPy is a sort of a standard matrix library in Python, and it's so common and everyone uses it that and people get really, really lazy. They don't want to write out the whole word NumPy. And so this is just sort of saying, hey, I'm just going to use NP to refer to NumPy. But that's sort of a pretty universal Python programming custom. And so what I want is I want a big matrix of zeros, and the size of that matrix is it's 1 by 100. And okay. So that's just saying, hey, I have a big giant weight matrix that is all zeros, but it's of the right size, and that's going to in that connection there. Now that I have that, if I right-click on output, I can also make a value plot there. And what we should, of course, see right now, because that weight is all zeros, is that if I run that, the output just stays at 0 all the time because it's just, multiplying all that activity by 0. So, of course, it's 0. Okay. So now we need to introduce this learning. Okay, there's two things that we're going to need to do in order to introduce a learning rule. One thing is to tell this connection, hey, there's learning on this connection. And the other thing is we've got to say, well, what is the error signal that is driving this learning? Okay. So let's start with that error signal. All right, so something is going to need to be computing some sort of signal that says, hey, is my output too big or too small? In some learning situations, this might be a signal that's coming in from your external environment. In lots of other situations, it might be something that the model itself is going to need to compute. So you might have a bunch of other neurons that are going to be computing this error signal. The syntax is going to be similar. What we're going to do, since we haven't even introduced enough of Nengo to show really how to compute things yet, what we're going to do is we're just going to do this without neurons. We're just going to assume that there is some sort of error signal coming in from the outside world. And we're going to call this error. It's going to be a node. And interestingly, we're going to say it's also a node that does nothing. Okay, and we're going to see why in a moment. Because really what we want this error to be is this error should be this output minus this input. Right? That would be the correct error. If I'm trying to learn to be able to output whatever my input was, then my error signal should be the difference between the output and my input. Make some sense? So if I want to compute the difference between the output and my input, well, I can do that with these Nengo connection things. I can make a connection from the output to the error. I can make a connection from the stimulus to the error. But then when I make the connection from the stimulus to the error, I want to multiply that by -1. And then if both of those are going into the error, then now the error, this error node should be the difference between the output and the stimulus. Make sense? So let's do that. I'm going to make a connection from the output to the error. This is also sort of an example of programming with neurons and with these sorts of neural connection type things can look a lot different than normal programming. In normal programming, I would just say, oh, just subtract these two numbers. But here I've got to actually say how this is being done. So I'm going to make a connection from the stimulus to the error. When I just make a connection and I don't specify anything, there is a default transform that's being assumed, and that's the identity matrix. So just multiply by 1. That's not what I want in this case. I want in this one, I want the transform to be -1. Okay. Yes, you do want to make sure that you get the sign on that right, that then it's an error. So if the output is larger than it should be, then that should be a positive error. So that's why it's output minus the stimulus. Let's run that and make sure that's doing what we expect. So I'm going to pull up a value for the error. Okay, so all right, if I feed in 0.5, the output is 0 because all these connection weights are 0. And the error is around -0.5, right? Because I'm lower than I should be. I'm outputting 0 when I should be outputting 0.46. If my input is, I don't know, -1, my output is 0, my error is 1, because my output is 1 too high, right? So we are successfully computing the error. Now I need to actually use that error to modify my weights. How am I going to do that? So first thing we need to do is this connection, we need to indicate that, hey, this is a connection that has a learning rule. There are a bunch of different learning rule types in Nengo. So the particular learning rule type that we're using right for this example is the prescribed error sensitivity rule. Oops, let me adjust my screen resolution size a little bit so we can see that. Okay. So that's how we're saying this connection has a learning rule on it. There's a couple different parameters for the error sensitivity rule. The main one is the learning rate. So let's toss that in as well. Learning rate 0.0001. Okay, and let's just for clarity bring all that over there. Okay, yeah, let's indent that a little bit. There we are. That's fine. Don't want to take up too much of my screen with the code. So now we have a learning rule on that connection, but now we actually need to connect our error to that connection in order to say that that error is what's controlling this learning. Right now, when we've defined this connection, we've just sort of created it, but we didn't keep a reference to it. We didn't assign a variable name to it. So at the moment, we can't actually refer to this connection. We're going to fix that. I'm going to say, all right, C is that connection. And then because I'm a little bit obsessive, we're going to go fix our indentation a little bit. Here we are. So C is this connection, and now that I have all of that, now I can come down here and I can make a connection from error to C's learning rule. To C's learning rule. And you will notice that that now shows up as this sort of green dotted line saying that this error is now going to be modifying this, the weights on this connection. Cool, all right. Is that going to work, or did I mess something up? Let's find out. I'm going to save this. Sure. Press play. All right, so we've started off. I've just paused the simulation here. We're feeding in an input of 1. The output was initially 0. The weights are being changed. The weights are being changed to sort of, and our output is gradually growing. And we should level off at an output of 1 and an error of 0. Of course, we've only fed in one input now. If I start moving this input up and down, right, it's got to learn across this whole space of things. And if we do that a little bit, you will now notice that the error starts to stay pretty constant. Well, sort of near, well, not quite constant. Right. Now, this is, of course, going to get tiring of me moving my mouse cursor up and down. So let's also use this as an opportunity to introduce, I would just like to have this input vary up and down on its own. Right. So basically on this stimulus, instead of setting it to a constant 1, I can define a function. So we call it stimfunc. It's a function of time. And I'm just going to say return the sine of t. So yay, let's feed in a sine wave, and then the stimulus is going to use that function. I run that. Oh, and this is also perhaps an opportunity to speed up time a little bit. Here we are, so the slider here is going to get things to run as fast as it can. And what we should now start seeing is that it has learned to keep the error pretty darn small across that whole range. So in that sort of few seconds, it has gone ahead and found a set of connection weights, just using that delta rule, just using that sort of gradient descent, or however you want to refer to that, just this prescribed error rule is just adjusting these weights, and is now doing a pretty good job of keeping the actual output pretty close to the desired input. Right? So this is now a system that has learned to what that set of connection weights is. So it's a fairly simple thing to learn. Indeed, that should sort of converge to the same answer that Nengo gets if you just solve when you solve for it. It's using it should end up with the same set of weights that Nengo found by solving.
Thanks, Terry. So what you basically did there was show the learning rule, and give an example of how we can do online learning in Nengo, putting a little bit of infrastructure in order to generate the error signal. But once you do, then sort of a fairly simple matter of introducing the actual learning. Or as you mentioned, every time you hit that play button, you see a little gear going and doing some thinking. And what it's doing in that instance is essentially finding those decoders, these DIs it is. And as you mentioned, it's solving an optimization problem. And so just to be a little bit more explicit, it's actually a fairly straightforward optimization problem, really. All you're doing is saying, I want to make my X be exactly the same as my X hat, where X hat is my estimate. You'll remember that X hat was equal to the sum over all the neurons of the activity of the neurons times these decoders. So really what we're saying is over the space of inputs, so in your case, you explicitly went and made sine waves that went from -1 to 1. For our optimization problem, we would just say, for every value between -1 and 1, we want to make sure that the squared difference between X, our sort of target, and X hat, our estimate, is going to be equal to 0, right? We want to minimize this function. So just a couple of notes on notation. We're using these angle brackets to indicate integration. We're integrating over x. So this is telling us what the range of the integration is. And we're going to minimize the function such that this is basically 0, if possible, as close to 0 as possible. And the thing that we can manipulate are these d's, right? So we would essentially plug this into here, and then, take the derivative of that error with respect to these d's and minimize it. This turns out to be a standard least squares problem. So there are some really nice, efficient methods for finding those d's that we can guarantee are optimal, because it's a quadratic function that we're basically minimizing here. And that's really what the math is that's going on when you hit your button and you see that little gear spinning away. We also tend to find that this is way faster to do this optimization problem than it is to set up all the learning and have all that learning happen. So unsurprisingly, we tend to build models and just let Nengo find those decoders for us, unless we're trying to actually explicitly ask questions about how could those connections be found online at runtime. All right, so another way of just sort of explaining exactly what's going on here and seeing the difference between one neuron or two neurons and more neurons, I've got this nice graph, which on the left, we've got a picture of the firing rate. So these are just the firing rate curves, exactly like the ones you were plotting in Nengo in the details menu. And on the right-hand side, we have what the DIs that would be found optimally, minimizing that error function, would look like if we plot it. In this case, we're sort of ignoring time. We're just pretending everything's rate and we've got infinite amount of time and so on. So with two neurons, we can get an estimate which is okay. The dotted line here, this dotted line is indicating what the perfect solution would be, right? So for an input of -1, we'd be outputting -1 if we lay on that line, and so on across all the values between -1 and 1. But when we're weighting the curves, they basically turn into these two things. So this is our off neuron being weighted. This is our on neuron being weighted. And if we sum them, we get something where we have a couple of kinks and so on. And those kinks are introducing a little bit of error. And so that's, as good as we can do with two neurons.
That's also, we saw exactly that effect in the example, the two neuron example I was doing, where when I fed in a 1, I was actually getting a value around 0.8. So it was this, exactly the same kinks were happening.
Yeah, so you can see there's a little bit of error here. And you also saw that actually estimates were better in the middle because both neurons were firing, right? Yeah, so a lot of that did show up in exactly that example. So now if we start picking things randomly, so here again, here we've got the ideal Alice and Bob where we've picked both the intercepts and the maximum firing rates quite closely. Here we're starting to do just random stuff, right? So now we're just going to pick neurons randomly. And we picked 8 neurons. And so we can see this is, for this particular set of 8 random neurons, this is how good we get. And we still see a little bit of error up at the top here. So we can increase the number of neurons. And then for 30 neurons, we now start to see, wow, that's actually, pretty good for only 30 neurons. Don't forget, brains have billions of them. So we could be doing really well. But of course, we also think brains are doing much more interesting things than just trying to represent their inputs. But nevertheless, this gives us a sort of sense of how this quality of the representation is improving as we add more neurons. Or similarly, the root mean squared error is going down, right, as we add more neurons. So that's good because, different inputs might need different quality of representation. So if we really want to precisely know the location of that fly we were trying to catch earlier, we might need a fair number of neurons dedicated to it. But if we only need to know if it's on the left-hand side or the right-hand side of our visual field, then maybe we don't need so many neurons. Now, we can start doing something more interesting than sticking in a single dimension. So all the examples we've seen so far are these sine waves that we've been putting in. And what we'd really like to do is have a much more general purpose representation for a vector space. And ultimately, we want that vector space to potentially be really high dimensional. But let's just take the first step of adding a single dimension. So what we're doing now is representing a sine and a cosine. So that's these two inputs over here. We can think of that in state space. So you'll notice that we've got sort of time running along this axis here, which looks like all of the plots that we've seen in Nengo so far. But we'll see that we can also have plots over state space where time is going to be essentially implicit in the fact that we're watching a video, right? And so on the static image, we've represented time with this arrow, where the input is kind of cycling around across this input space. And what I'm showing with each of these now is a more sophisticated notion of an encoder. So as you mentioned, Terry, and as I had mentioned, when we just have two neurons, or rather when we just have a scalar input space, for any number of neurons, we only have a +1 encoder or a -1 encoder. That's essentially a one-dimensional circle. If we have a two-dimensional circle, now we can actually pick an infinite number of encoders, right? Because for every possible direction, right, we could define an encoder for a neuron. So now our E's that we had before, so you might remember we had this equation AI is equal to GI alpha E times X. These encoders are going to be vectors because we're representing a vector space, a 2D space now. And in a 2D space, an encoder cannot just be +1 and -1, but rather it can be any combination of numbers which result in a unit length vector pointing in some direction. And so that's what the set of encoders is going to be. In the example I've got here, we've just got 4 neurons. So I've numbered them 1 to 4 in this diagram down here, where for each neuron I've picked one encoder going at 45 degrees in one of the quadrants. And then we can see if we have those 4 encoders, so these are the 4 encoders, and we use this particular sine cosine input, meaning that we're moving around in a circle in the state space, so we're moving around the unit circle. Then over here we see what the spike trains should look like. So here you can basically see that at this point we're passing by the 45-degree angle of this in this particular quadrant, which is making that neuron fire the most because it has its tuning curve pointing in that direction, right? And then we can see neuron number 2, we can say, look, now it's pointing down in this direction, and then neuron 3 and neuron 4. And so we can see at different points in time that we're basically getting to different parts of the state space. Now, the other question that we often get for the NEF is, we always start by introducing, as we've done so far, tuning curves that look like this, where we've got, X is increasing and the firing rate is increasing or decreasing. And people are often a little bit puzzled by that because, if you go to neuroscience and somebody shows you a tuning curve, probably the most common tuning curve you'll see is one that looks more like a bump function of some kind. And, this could be like the orientation of a bar that you're sticking into a cell in V1. It could be the movement direction of the arm recorded from motor cortex. In all of these cases, we see these tuning curves that have bumps. And so the question often arises, well, how do you get bump tuning curves in the NEF when you've sort of got these linear functions that are increasing all the time? And the answer is basically if we plot our 2-dimensional space, or n+1 dimensional space, in n dimensions, in this case in 1 dimension, we'll tend to see much more interesting shapes than planes or sort of slightly curved planes like the one that we're seeing here. And that's what this last graph is showing here. So this is showing the 1-dimensional tuning of those 4 different neurons. Even though they're defined in a 2-dimensional space, we're plotting as a function of theta down here, right? So this is showing exactly how far around the circle we are. So again, we can see for this neuron, when we're this far around the circle at 45 degrees, it's going to have its peak firing. And that, of course, corresponds to this peak firing in time, because that's where we are at the 45-degree point in our input on the unit circle. So I think this is always a nice thing to point out. It's kind of quick in some ways. People are like, I'm just trying to understand what you mean by the 1-dimensional tuning, and now we're in 2D, what's going on? I think the important thing to note is that nothing about those equations we've shown change, right? So all that really happens is the encoder is now 2-dimensional. The decoder is going to be pretty much the same too, where we have an X hat, which is now a 2-dimensional space. That's also going to be a sum over all the neurons. We just take all the activities of all those neurons, we multiply them by some decoder D. And as you can imagine, that decoder is now also going to have to be 2-dimensional. So this is going to also be a vector. And we're taking all these 1-dimensional activities, we're multiplying them by a bunch of 2-dimensional decoders, And that means after we sum them all together, we're going to get a two-dimensional estimate. And so really everything's the same from a mathematical perspective. We started talking about vector spaces, and then we just gave a bunch of examples where a vector space was one-dimensional, it was just a scalar. But the math stays the same. When we move to 2 dimensions, a couple of interesting things happen. One is that our encoders now become, there's an infinite number of them. We could pick a whole ton of those instead of just plus and minus 1. Another is that if we start plotting our tuning curves in a dimension other than the one that we're actually sort of defining our neurons in, we can get much more interesting kind of tuning shapes that people are familiar with from looking at neuroscience results and neuroscience data. But other than that, from the sort of perspective of the math and the representation principle, things are really pretty much the same. So we have the same encoding and we have the same decoding. So I think with that, I will turn it back over to you, and we can see how straightforward it is in Nengo to add more dimensions.
Cool. Yep. Yeah. And certainly that this adding more dimensions, it takes a while to get one's head around it. And but for me, like the take-home message is, if you've got a particular neural data that you're wanting to make your neurons respond like, then it's just a matter of choosing your represented space X and your encoders, and you can always find some way of organizing that such that it'll get you whatever it is you're seeing in the biology. I want to say always. All the times that I've ever done for whatever neuroscience data I've ever looked at, it's been straightforward. Yeah,
I think, exactly right. Every time we've tried, even for really weird, you have 2 bumps in your tuning curve. How do I get that? It's like, yes, you just, add a couple of encoders together and off you go. Things work out.
All right, so the example that we have built up with all the learning, we're just going to save that one and start a new one and basically throw everything out and start over again. But we're going to go now to example 4. And yeah, let's go ahead and just wipe all this stuff out. Okay, so we have, right, so our whole point here is we want to represent 2 dimensions with our neurons. So, and we're going to say is, eventually I will learn how to type, ensemble. We always have to specify the number of neurons. I'm going to do 100 neurons here. And the dimensions is now those 100 neurons are going to be working together to represent 2 things. Okay, an X location and a Y location. Great. So that's a group of neurons. We're going to we need some sort of stimulus to go feed into that. And now our stimulus, though, is going to be multidimensional. So we're now going to say 0, 0. And now we have a stimulus over here that can be multidimensional. And of course, we have to connect them up. And go connection stimulus to A. All right. I'm going to close all these graphs that might have popped up for you, just to sort of start fresh. We have a slider that we're going to pop. Now the slider, you'll notice, now has 2 sliders because we have 2 different values we want to go modify. And for right now, I'm just going to right-click on A and look at the spikes. Okay. Maybe make that a little bigger. Here we have neurons that are responsive to randomly chosen directions in the unit circle. So there's sometimes it's called preferred direction vectors or preferred stimuli. Each neuron has some preferred combination of these 2 inputs. So it's not like half the neurons are being affected by one of these and the other half are being affected by the other. So you'll notice if I move this slider, all of the neurons are being affected by this slider to varying degrees. Some of them are being affected a small amount, some of them a large amount. Right. And same with this other slider. Right. All of those neurons are being affected, some more than others. Right. The other one way you can think of that as well is if you were sort of imagining this as a normal neural network. If you had like 100 neurons here and 2 inputs, then you would have a 100 by 2 weight matrix in here. Aspects, so that weight matrix you can also think of as that's sort of like the encoders. technically it's the encoders combined with the gain scaling factor, but the idea that there's something about these input, this sort of the connection weights going from these 2 to these 100 things, that's what's giving us this diversity of neural response. Mathematically, it's the same thing that we were just talking about. But it's a little bit hard to see with this plot. And so that's why I'm going to pull up this value plot. So the value plot is the take this spiking activity, do the decoding of the spiking activity. And in particular, we now have 2 different outputs. Okay. So you can either think of that as the decoder is 2-dimensional, or you can think of it as there's one set of weight, there's one weighting of these spikes that if you add together those spikes and do one particular weighted sum of them, you'll get the green line, and the other weighted sum will get the blue line. So one weighted sum will give you one of these inputs, and the other one will give you the other one. Same thing. Okay. Again, a little hard to visualize this way, so I'm going to do one more plot. Now that we're into multidimensional, we now have a new option in here. This is the XY value plot. Right, and this is going to be plot the same data as on this thing, but plot it as a state space diagram. And I'm going to do the same thing for the stimulus as well. XY plot. So same data. All right, but now what I can do is I can sort of say, all right, so this little blue dot, that's where my input is. My output is similar spot. Slow down time again. You will sometimes find in your display that the spike plot starts lagging behind. If that does happen for you, slow down your simulation a bit. Basically what's happening is the browser isn't getting enough time to redraw the visualization. So this slider down here should be controlling how quickly your simulation runs. If you run it at full speed, it can sometimes mess up the display. Pull it back a little bit. Anyway, back to what I was saying. This is the decoded information. And what we should end up seeing is we might have one particular neuron. Where's a particular neuron? Can I even find one? That is, so this neuron here, the third one from the top, is very sensitive to this part of the space. It's not sensitive at all to this part of the space. It's not, so this, again, this third neuron is not firing at all in this part of the space. If we go back here, it is sensitive to this part of the space. So this neuron is sensitive to this sort of part of the space. So it's got an encoder in this direction. Right, whatever direction causes the maximum firing. And again, that's something that you see commonly in biology. You get this sort of uniform distribution of preferred direction across the space. See, that's, yep. And again, you can generalize this. You can go up to 3 dimensions, you go to 4 dimensions, 5 dimensions. We've done models 512 dimensions, I think 800 dimensions. If we, the math all works out the same. Of course, as you increase the number of dimensions, if you have the same number of neurons representing more dimensions, you're going to have interesting reduction in accuracy.
So as Terry was suggesting there, we can actually begin to build up a kind of hierarchy of representations. So we started with 1 dimension. This is a simple case. So the example of the goldfish eye movement in the horizontal dimension, that's kind of like a 1-dimensional representation. In the horizontal axis, the eye is located. And we know that there are neurons that are sensitive to that and so on. So that's the 1-dimensional example that we can find in biology that we sort of started with in introducing the first principle of the NEF. And then we moved to a 2-dimensional example where we were moving both in the X and Y directions. When you have animals move their arm in a sort of constrained 2-dimensional environment, so you basically give them a joystick and they can move in one of 8 directions. There's a whole ton of experiments out of the Georgopoulos lab, which looks at representations of this variety. They're very well characterized in terms of this 2D representation where they have preferred movement directions, which are captured by these two-dimensional encoders. And we can represent those quite nicely by picking those encoders randomly from a unit circle, and then sort of decoding and getting an estimate of the output. We can keep adding dimensions. We can add 3, and then we get up to something that looks kind of like the semicircular canals. So these are 3 perceptual sensors that we have in our inner ear that determine head acceleration and velocity in the 3 different dimensions. And they tend to be oriented pretty much in the cardinal dimensions that we would expect, so horizontally, vertically, and in the forward direction, in order to detect our head movements and be able to inform our body about balance and so on. So it's quite natural to think of the neurons associated with each of those canals as having a sensitivity in this 3-dimensional space. And we can, of course, decode 3-dimensional head acceleration by looking at the activities across all of those neurons. We can begin to generalize this beyond sort of a number of dimensions up into infinite dimensional spaces. Well, what do I mean by that? Well, technically a function, so a one-dimensional function, can be thought of as an infinite dimensional space because it's continuous. So if we say, the direction theta that the head is pointing in the hippocampus, as represented by hippocampus as a function, then we can represent that function by defining encoders over the functional space, these EIs. We still have an activity also defined over that space. And we can use exactly the same mathematics where we define an encoding and a decoding. The interesting thing here is that for any infinite dimensional space, like any function like this, really we can't represent an infinite number of points in that space. So ultimately we do some kind of decomposition to represent that with some degree of precision. So in the ideal case, we might want to be representing a perfect function that looks like that, where the head direction is exactly equal to that particular direction. But when we record from hippocampus, we notice that neurons are tuned sort of broadly, and if we look across all the neurons, we see a function spread out like this, which is really an f of y. But we can't move an arbitrarily small amount in that space and see that representation updated perfectly, meaning that there isn't an infinite amount of precision in the representation of this potentially infinite dimensional space. So instead, we can start thinking about functional decompositions, right? So the most common one of these is the Fourier decomposition, where we represent arbitrary functions. So some arbitrary function f of x is going to be equal to a sum over a bunch of coefficients a times my basis functions in the Fourier space, right? And if we start thinking in those terms, then it's these a's that we really have to pay attention to, and then they become finite dimensional, right? However many Fourier components you have, that's the number of these coefficients you have to represent, which is the number of dimensions you have to represent back in a vector space. So I'm sort of introducing this notion, and we're not actually going to dive into it in too much detail. As with everything we mentioned, if you want more detail, you can definitely check out the book or a bunch of our publications. But I think it's nice to think of in terms of a representational hierarchy where we're really going up in a sort of representational complexity as we add dimensions, and we can start thinking beyond sort of finite dimensional spaces and think more in terms of functions when it maps well onto what we see biology doing. We can push this even farther, go into an x-dimensional function. So, a visual image we can think of as a 2D function where the intensity at every point in that 2D space is, the intensity of light hitting our eye from that point in space. More than just the intensity of light, we can also define other properties at every point, like the depth. So we can have, more than one thing defined for each point in the function. So then we have a sort of x-dimensional or an n-dimensional function. Depending on the number of properties we define. So we can define color and intensity and depth and all these things at the same point. And sort of leaves it open to the imagination of the modeler or the neuroscientist. What kind of representations do we think are important to be injecting into our network? And conveniently, the mathematics stays sort of systematically the same, which is one thing that we like about the neural engineering framework and each of these principles. And we'll start into the next lecture by beginning with an example and beginning to look at how do we introduce computation into the models and how we begin to have layers of neurons and connect those layers and have them do those computations in some sort of interesting context. So yeah, thanks.
Sounds great. Thank you, Chris. Thank you all for listening, and we'll see you next time.